In the beginning of meiosis, DNA strands thicken into chromosomes. Homologous chromosomes begin to connect to each other. The centrioles divide and move to opposite poles of the cell. This is where the chromosomes cross over. In metaphase 1, the tetrads attach to spindle fibers at their centromeres, and they line up mid-cell. In early anaphase 1, the tetrads separate, and the paired chromatids move along the spindles to their respective centrioles. In late anaphase 1, the chromatids have almost reached the spindle poles, and the cell membrane begins to separate. In telophase 2, nuclear membranes enclose the separated chromatids, and the cell membrane completes its construction. The first division ends, and there are now two cells, each with the same number of chromatids as the parent cell. Now prophase 2 begins. In the second division, homologous chromatids do not duplicate but separate. Metaphase 2, the chromatids line up in the middle of the cell. The centrioles and asters are at the poles, and a spindle has formed. In anaphase 2, the separated chromatids approach their respective poles, and the cell membrane begins to separate. Telophase 2 has been completed, and there are now 4 cells, each with half the number of chromosomes of the parent cell. In interphase, or G2, the cell grows. In the S phase, or early prophase, new DNA is synthesized when chromosomes are replicated. In late prophase, the centrioles go to the opposite poles and chromatodes form. During metaphase, the chromatids line up in the middle of the cell. When anaphase begins, the cell starts to split into 2 separate cells. In telophase, the cell finally splits into 2 separate cells. Finally, in interphase, or G2, 2 cells have formed, finally completing mitosis.